PSS30 folate transporter. Folate transporter manuscript is also under consideration for publication. So then 62125, they are being written. So what we did in the meantime is that after cloning these genes, we transformed soybean with all these 5 genes using 3 different promoters, and then so that each of the genes can confer SDS resistance, as well as 2 of them, 25 and 30, can confer SCN resistance. So that's where we are. Then we are starting to use these transgenes to stack, which I will show you a slide some point soon. And then basically, we are also focusing on studying the mechanism of PSS1. We have shown that PSS1 is probably, PSS1 is involved in regulating the autophagy for causing the or conferring the non-host resistance. So my graduate student, Toyo, is working on this area. So then this is the presentation showing the application of transcriptomics in cloning important genes that can enhance disease resistance, and also then at the end showing that how we can use transcriptomics to identify the possible mechanism used by the soybean gene in conferring broad spectrum disease resistance. Without further delay, so oh, I guess we already seen this, and I think I am going to skip this one. Yeah. So what we have seen here is that 4 classes. This is the work conducted by, initiated by Binod Sahu, who started my lab some time ago. I forgot exact date. I don't want to guess. Probably it was in 2008 or so, I think, after he finishing his first postdoc. And then he did this interaction study, and then we did the sequencing, deep sequencing of transcripts from control and infected hypocotyls or young roots, of etiolated seedlings, so that we can get better infection. And we took 10 days and 24 days, maybe it's all details in the paper, and my memory is now not doing well. So, but early and late time point. From that, what we found is that only like 20 genes were suppressed or downregulated by infection, and they can be classified into 4 groups. And you can see that one representative sample from each of the classes, 1, 2, 3, 4 here, that is in control early and late. So you can see in control, they continue to transcribe. There is no problem. But as soon as you put the infection, then they rapidly decline. So barely you can detect those transcripts. So this is basically reads from the experiment of transcriptomics. You sequence the cDNA prepared from control and infected tissues. Sorry, yeah. So the 4 representative samples from 4 classes of genes are shown here. These are the 4 genes that are shown in the previous slide. And one encodes CIV element occlusion protein, the other one ankyrin repeat containing protein, SAM dependent carboxyl methyl transferase, and disease resistance 1 that we named because this was a novel gene we started, and nothing was known. And so to validate, we conducted RT-PCR to show that this is the early point, because before early point was like 10 days. Now we have done 8 hours to 5 days. As you can see that this is the ELF1B, is a constitutive genes expressed everywhere. Then here you can see that there is expression in the control, but as soon as you infect, the level of transcript goes down very clearly. And here in this case, this is the novel gene GMDR1. there is no transcript left at all as soon as you infect. So then it is very obvious. Now you may ask me why we focused on susceptible or pathogen suppressed genes, because I think general trend has been in the past to focus mostly on um, genes that are rapidly expressed following infection. The reason is that we hypothesize that maybe some of the genes are defense related and maybe pathogen suppress the expression of those genes, so then it can cause sudden death syndrome in soybean. So with that hypothesis, we decided that how about we change the promoters? Now, if pathogens are involved in suppressing the expression of these genes through promoter, it has to do it through promoter because it is regulated at transcription level. So we decided to create transgenic lines so that we can swap the promoters of these genes with 3 different promoters that are infection inducible. One is very specific to infection, 2 are root specific promoter, very strong. They also induced by infection. So in this diagram, we can see non-transgenic plants, the gene expression of these genes are suppressed. As a result, susceptibility is the outcome. When we change the promoters in transgenic plants, following infection, the expression of these genes will take place and then resistance would be expected. That is our hypothesis and working plan. And with that, we showed that when we do, then all these genes are able to, all but one. So first one is not showing any differences in the symptom development, but 3 of them anchoring repeat containing that is published in 2016. And this is in progress. And then also this one is published as well recently. So you can see that each of these 3 genes can confer SDS resistance, and also 2 of them can actually confer SCN resistance. So what we do now is that since we have resources that these 2 genes are good for conferring both SDS and SCN resistance, and earlier I showed you that 25 and 30 are involved in both can enhance SDS and SCN resistance. We are now putting these 4 genes into one plant and then asking the question whether there is what is called complementary effect, additive effect, so that there will be further enhancement in the SDS and SN resistance by these 4 genes. We have so far, so these are the 4 candidate genes used for gene stacking experiment. Basically, Ms. Nagaki is crossing these into one plant now. Segregants are being grown in the greenhouse, and she is, as we speak, she is starting to do PCR to identify lines that have homozygosity for all 4 genes. And once you have that, then we'll check the responses to both pathogens in next year. And in the meantime, she showed that the experiment actually works using 2 genes, GMDR1 and PSS30. Those 2 genes are incorporated into 2 independent lines. Both are homozygous, and this is the response of these lines to SCN. You can see the number of cyst development is reduced almost half of what we see in individual parents. So in other words, looks like it may be additive. The responses could be additive. Because all this mechanism of all these 4 genes are distinct. They are not of the same mechanism. They do not fall into the same pathway of disease resistance mechanism. So therefore, we hypothesize that stacking could be very beneficial. So today, I will focus mostly on characterization of GMDR1. And GMDR1 was, as I mentioned, it is a novel gene. When we started to work, nothing was known. It is actually also expressed only, it is only found only in legume species. And in addition to legume species, we also find homologs in jute, as well as cotton, and maybe poplar also. But we don't see these genes anywhere else. among the species that have been sequenced. So we this work has been published just last month in Plant Biotechnology Journal. So if you are interested to learn more about it, so that will be the place to look for. So what we found initially is that when you overexpress these genes in soybean, then we do see resistance shown in the middle. And this is the recipient line, Williams 82, and no resistance from DR1. And then this is the control resistant or resistance. It is resistant to Physium pargoliforme. Here is more to show you that we did 3 different promoter, 2, 3. There is an enhanced number of resistant plant. And here, Williams 82 recipient, you have not many. This may be escape, but you can see that enhancement of our days. These are plants that are proportion of the segregating population in transformant individual transformants using promoter 1, 2, and 3. And this expression of resistance is also associated with the expression of the transgene shown here. And in the field, we had to do because a resistance we see in the greenhouse or confiron is not always associated the resistance we see in the field in this particular disease. And therefore, we had to grow in the field for several years. And then you can see transgenics are doing well, but non-transgenics are infected by the pathogen. Again, this is summarizing all the results we have for this particular pathogen. So you can see this is already that. You can also see resistance in the roots. So this is the transgenic recipient plant, and this is the transgenic plant showing SDS resistance in the leaf, which is known as foliar resistance. And here you can see also root resistance, no rotting like here. And again, this is the proportion of roots that are rotting. So you can see reduced rotting here in the transgenic and as opposed to non-transgenic. And again, this is, again, showing that this expression of resistance is associated with the expression of the gene and not in the susceptible, there is not much. And this is the Williams 82 control. And here you can see that the amount of inoculum produced by the pathogen in the resistant roots are very little or less significantly lower than the one you saw in recipient plant. And in the field trial, we did these 4 different years, 15, 16, 17, 18. And each of the years, the amount of the severity of the symptoms were also very different. But as you can see, in each of the 4 years, we saw significant level of resistance among the transgenic lines as opposed to non-transgenic recipient Williams 82 line. So this is where the story becomes interesting. So one day, Michelin and I was walking through the greenhouse. We saw this plant, and then we started to notice because the rest of the plants, as you can see in the background, they are infested with spider mites. And so spider mites in the summertime is a severe problem in greenhouse. As you probably know, that spider mite resistance is very much not available across the plant species except for a few exceptions like tomatoes. I have seen reports, not many reports in other crop species and not in soybean. So this is striking to us. We thought that maybe it can be some clonal variation. As you know, when you grow plants from, in tissue culture going through, it can cause some mutations that we don't know. So, but maybe also from transient. So therefore, we decided to follow up on these plants, related plants with this DR1 also show the resistance. And I forgot to mention the background lines are from different transients, including non-host resistance PSS genes. And as you can see here that these are the mite to show you spider mite, and you can see non-transgenic in 2 weeks and 2 months, and transgenic 2 weeks and 2 months. As you can see that this difference is very obvious. We see single gene segregation in self-generation from this transgenic mutant shown here. So we are convinced that this is not just a fake. It is by DR1. That was quite striking. We did some more work to quantify the amount of resistance using some leaf assay. The results are shown here. You can see that the number of spider mite eggs produced in the resistant plants are only 4 to 8, whereas here you can see 20. So there is a tremendous amount of reduction in the development of chlorosis. As you can see here, chlorosis is defined as the amount of chlorophyll by measuring the chlorophyll. You can see lots of chlorophyll here, transgenic, but there is a reduction in the control. And here we just saw the expression of the transgene. Wherever there is a resistance, then we see expression. Red one is not resistant, you don't have expression, and so it's controlled. And these are endogenous genes that initially there was some expression, but day 3, I think the expression is reduced significantly. This is then, since it expressed mite resistance, we also ask the question if these plants can confer resistance to aphids. And we did see resistance to aphids, which is a major problem in soybean as well. Again, showing the graph here, you can see significant level of reduction in the aphids per nymphs in the leaf surface as opposed to recipient Williams 82. Again, it is associated with the expression of the band, which is, these are the resistant, aphid resistant plant. They always produce the transient expression. Now, we have seen that the plants are good. So therefore, we decided that maybe it will confer SCN resistance because it's a very important pathogen. So we did that too. And you can see that this is the control, non-transgenic. You have a lot of, these are called cyst, female. After completing the cycle in 30 days, then it forms a cyst inside which there are a lot of eggs and that are released to the environment for making hundreds of eggs. So that will produce the juvenile and have a second cycle of infection. So then you can see in the transgenic line, there are not many, there are 1, 2, 3, 4, 5 maybe. And so again, the female index is shown, which is the proportion of eggs. You can see very few here as opposed to non-transgenic transgenic recipient Williams 82. Here you can see that as compared to that, we have very few showing that SCN resistance. Again, that is associated with the expression of the transgene. And these are the non-susceptible, SCN susceptible transgenic and recipient plants shown here. Then we were interested to understand where the protein goes because it gives resistance to 4 different pest and pathogens. So maybe a receptor like protein or something. So, and it is only 73 amino acid long. We did some bioinformatics analysis of this. We can see that there are 2 transmembrane domain underlined here. There is aspartic residues for cleavage of this peptide right here. This is a serine residue. If it is involved in phosphorylation, we don't know, but statistically it shows that it can be. Then this is the cytoplasmic domain, and this is a 3 amino acid, maybe membrane bound or cytoplasmic. So it's basically a tiny protein, and it has the power to show its power when you overexpress it. So tiny, but when you overexpress it, it can be very useful. And we don't see any deleterious effects on the transgenic lines under field growing conditions and or in the greenhouse. So we don't see any side effects of this transgene, although we have not evaluated other than just ill for other traits, but we do have some data on transcriptomics, so which is kind of very encouraging. So here is the confirmation that it does go to the plasma membrane, showing that protein goes to in the membrane, confirmed with the internal control, which is also plasma membrane protein shown to be. And when you merge these 2 pictures, then you can see that orange color indicating that both proteins are going to the same location. Since this goes to plasma membrane, therefore our protein goes to plasma membrane also. Then we confirm that it is not cell wall protein because plasma membrane and cell wall are side by side. So therefore we deplasmalize the cell, and using sodium chloride, you can see that now this is going along with the protoplasm here. And so is the plasma membrane protein internal control. Again, when you merge, you can see clear alignment of both proteins along with this membrane fraction, which is collapsed due to plasmolysis using sodium chloride. So it is now next phase of the work. So now we have it. Now what does it mean in terms of mechanisms? How does it work? But one thing for sure, I want to re-emphasize that there is no natural mite resistance in soybean, and therefore it is a agronomically important trait we have created through biotechnology. And that's why I think transformation experiments are important, although we have so much negative feeling about it, GMO across the globe. But I think that this is funny, so it came to my mind. So when we are working on it, there are so much negative GMO. So sometimes I feel whether I should be going to conventional approach and forget about this because it is not going to go to farmers' field. Then I realized that, well, I just do my job. Who knows? Maybe there will be a time when use of this transgenic material will be much easier in the farmers' field when we have calamities like, famine. I hope not. It should not happen. But but if we have the resources, always it can be usable. And mite resistance is one case where we have created through our expression of this protein. And also, this is also interesting that this protein is actually suppressed during infection, but that means what does it do? Is it really, it has this function to protect the plants to begin with, then pathogen has adapted to suppress it because it is a master regulator of disease resistance. And those are the questions always in my mind. And so then how does it do it? How does it provide the broad spectrum disease resistance? Then we ask the question that what can be common to all poor pathogen and pest, and it is the chitin. So chitin is a small molecules, and it is has been shown to be elisters or pathogen-associated molecular pattern that can induce PTI or PAMP-induced resistance, commonly known as also basal resistance. So then, therefore, to understand more about the mechanism used by this, we applied transcriptomic approach. Again, I will show you the power of transcriptomic approach in understanding the mechanism of this gene, how it does the how it confers the broad spectrum disease resistance and pest resistance. Before I do, this is the resources we published in this paper again. So what we found is that these are root-specific promoter, but still they do express in leaves. And this experiment is to show the level of expression in leaves because we found resistance to all these pests and pathogens. So obviously, although it was defined as root-specific resistance, in our case, we do see transgenes are highly expressed. So this is GMDR1. Then you don't see any expression because this is transgene. These are non-transgenic. For GMDR1, gas is a transgenic, but it's not for GMDR1, no level. But you do see about 250-fold of the endogenous gene. And then you do see it here, 1700-fold increase. And if you compare that expression level with the native endogenous CMDR1, it is only 1-fold. So there are three more homologs. There are a total of 4. HMDR4 doesn't express. That's why it is not here. But there is also low level of expression of DR2 and DR3. As you can see that expression of this transgene is not affecting the level of expression of this endogenous gene. So there is no role of these genes in our transgenic studies for conferring broad spectrum disease resistance. Important point to emphasize here is that the level of expression is wide here, 200-fold or so, 250-fold here, 1750-fold here. So we selected these 2 lines for transcriptomics. But remember that both lines can confer SDS and SCN resistance. So that's important. even low level, it can confer SDS and SCN resistance. So we named as weak. 136 is the line number. Strong 107 is the strong line. And this is the general protocols showing that how we did the experiment. We use strong, weak, and then transgene recipient Williams-82. These are the three genotypes used to treat with chitin or buffer, and then harvested 12 and 24. And then we did high-seq, all those sequencing, and then analyze the data. So our analysis has been completed for 12 hours, 24 hours, the level of transcription goes down, but that part is yet to be done. But 12 hours is the point when you do see a lot of activity or expression level. Here is the summary slide to show you the chitin treatment effect of transgene on the expression of chitin-induced genes. So this is a complex slide. So I'm going to take my little bit of time to explain to you. So you can see that you have chitin 136 is the weak, and Williams-82 is the transgene recipient plant. What we did was we identified all the chitin-induced genes in this plant, chitin 136 as well as Williams-82, and then asked the question is that among those genes which are log 1.5-fold across Williams-82 chitin treatment versus buffer, we listed those genes which are shown to be, that's then we compared those genes with chitin treated with 136. Basically, what we are asking is that, okay, we treated chitin transgenic and control, and which are the genes that are induced significantly of our Williams-82? That's the question we ask. Similarly, we ask the question that, so 107 is the strong transgenic, so treated with chitin. Williams-82 is treated with chitin. With how many genes are induced significantly of our Williams-82 treated with chitin? So 6,000 genes. So you can see that there are six-fold differences between the weak and the strong. And on the buffer side, you can see that buffer strong has 12,000 genes and only 400 genes in case of weak transgenics. Now, we cannot work on all the genes, it's too difficult. So what we decided to do is that since both weak and strong transgenes provide SCS and SCN resistance enhancement, so therefore what are the genes? How many are genes that are common to both weak and strong plants? We only found 289 genes. And on the other hand, for buffer, there are only 143 genes that are common to both. Skipping all the other genes, we focused on these 143 and 289. So then next step, we analyze these genes again, FPKM values, and then only selected genes that are twofold induced in 107 versus 82. These twofold changes either of our expression or, twofold increase or twofold decrease. We selected only the genes that are twofold of our Williams-82, reducing some of the background, and same here, same here, same here. So the results are shown here in a heat map. So this is also summarizing a lot of information. So let me start here. So first of all, we pull out the genes and expression level of in Williams-82 that are treated with chitin and has significant increase, twofold increase in the weak and strong lines. So basically the genes that have twofold induction in the weak and strong lines are only selected in here. And I think there are only 192 of those. You can see that if you compare the level of expression here, you can see that the level of expression is strong for this set of genes, 2/3. In 2/3 of the genes, basically you can see induction, strong induction, and 1/3 is suppression in the transgenic. So basically what I'm saying is that the little protein GMDR1 is involved in inducing this set of genes in the transgenics, whereas in this set of genes are suppressed. So this is kind of interesting. So then we did the same thing. The other thing is that we fill up this gap, we check those genes and then look at the transcript level in buffer control. So you can see that all these genes are not really affected by buffer treatment, at least in the strong plant, but there are some results here to show that there can be some induction in the but. So here is the results of the buffer. We found 115 from 143 that are having twofold differential expression as opposed to Williams 82 in both lines. We eliminated the lines that has twofold in one line, but not twofold on the other line. So that's what we did here as well to reduce the less probable line genes. So from this robust screening, we end up with 115 for buffer. What we see here is just opposite to here. Almost all the genes except for 2 or 3 that are suppressed by buffer treatment. So now what is going on? So buffer can cause damage to the cells. So we speculate that maybe these are regulation is due to DAMP, which is called damage associated molecular pattern. So maybe that is suppressing the expression of these genes in the transgenic lines. So further analysis is necessary. So to summarize, since I don't have too much time, so I decided to take a few key information that we found. One is 22 out of 192 genes. So these are 192 genes. So we focus on those. 22 genes encode leucine-rich repeat-containing signaling proteins. So that's kind of very interesting. So let me go forward one slide to show it more easily. Oh, so no, sorry. So then we took individual protein based on the annotation, and then we did also searching the individual protein for homology, and then we found that one gene is this gene is actually high identity to N gene shown here. About percentage identity is 35, but remember that this is soybean and tobacco is quite wide, and you see across the whole protein there is homology between the two proteins. This is another protein, this is resistant in Arabidopsis, known as NPR1 constitutive 1. Suppressor of NPR1 constitutive 1 is a disease resistant gene. So again, there is a high identity, about 32% identity, but this across the whole protein there is similar R identity. So then removing most of the background just to show you what that 22 genes are. There are 5 of them are containing only LRR domains. Then there are many of them, 4 or 5 of them with kinase domains and BSLR, leucine-rich repeat containing receptor like protein kinase family protein. And then there is one with NBARC and CC domain containing NBLRR, RP, W8. So these are 3 individual ones showing LRR containing protein with motifs for disease resistance genes. So probable candidate for disease resistance. And again, there is a quite a few 1, 2, 3, 4, 5, 6, 7, 8, 9 genes that are with tall interleukin receptor containing NBLRR protein there. There is a class of disease resistant genes. And you can see here that in soybean genome, 290 are CC and BSLR containing genes and 235. But there is a biasness in terms of induction of this class of protein in case of transgenic lines of are expressing GMDR1. So what does it mean? Then we also conducted MAN program to show it on the because these are only signaling proteins shown here. This is the biotic stress showing that these genes are part of the defense pathway, and like these are for auxin signaling, then ABA, ethylene. And so then there is also involved in protein degradation, proteolysis, cell wall protein. And here, this is a class that I just described. There are only few shown here, maybe 1, 2, 3, 4, 5, 6, 7, 8, 9, out of 22. So those are the signaling genes, and you can see that they are induced. and redox state regulation gene shown here. And there are also transcription factors and that are ERF1. Yeah. So in addition to that, we also found that there are certain genes that are related to abiotic stress. So this part is not developed well, which means that transgenic lines should be tested against resistance to drought or cold or other abiotic stresses. We haven't done that, but we did for salt. And we did see subtle phenotype on salt tolerance, but it needs require more work. In conclusion, I hope we are doing okay on time. Um, in conclusion, DR1 is most likely a PAM receptor that recognizes pathogen-associated molecular pattern and triggers PAM trigger immunity, commonly known as basal resistance. That's what we believe. Chitin could be one of the PAMs that are used by these 4 pathogens to induce the defense responses when we overexpress CMDR1. So now we have shown through transcriptomics that then there is a transcriptional activation of a large number of signaling genes that may be involved in biotic, maybe also abiotic stresses. One thing for sure is that not many R genes or NBL or R genes are transcriptionally activated, but this is an example where we have shown that master regulator GMDR1 is priming the defense response by overexpressing this in the plants, and also further inducing it following infection, which is kind of neat, something new. A large number of encoding kinases, LRR, TIR, and BSLR resistance proteins are involved in signaling and regulated by at the transcription level. So again, it is probably a novel trait because, as I have shown you that GMDR1 expression is really suppressed and we cannot expect any function following infection. Probably in the past, before long time ago, before pathogens were adapting on the soybean plants, maybe it was overexpressed and following infection, we don't know that. It may have a function, but now over the period of co-evolution of host and pathogens, pathogens were able to suppress this. That's another area to study. How does it do it? So we have to study how the regulation at the suppression or downregulation takes place by doing some interaction studies with pathogen protein and soybean DR1 promoter. That is another area to study. Then and the most importantly, what we are interested in currently is that what are the proteins that are involved in signaling. It is only 73 amino acid long, so it cannot do alone. So it has to be some kinases, receptor-like protein kinases may be associated during activation of this protein or the following infection. So that's part yet to be done. I hope I'm not taking too much time, or I have a slide to acknowledge the people. As I said, Devinder Sandhu did the mutation work, Minju developed the system, Binod Rishi did the screening work, Prasant Singh came along for characterizing some of the PSS since later on, so is Sekhar Kambakam, Bing and Saminathan, they all did field work for PSS genes. And Nisili Nagaki and Binod are in the second part of the talk on transcriptomics. They did an enormous amount of work. Michelin especially has conducted research on characterization of DR1, and currently she's doing the transcriptomics. Debbie Kandal, Yang, they did on non-host project. And her name is Toyo. She is a graduate student and she has been working on characterizing of PSS1, so showing that it is involved in autophagy. Michelin and Fefe and Subut. So we always meet once a week, Friday from 5 to 6 to discuss about transcriptomic works and in a collaborative pattern. So and grants. Iowa Soybean Association is the key consistent. I have been here 20 years. Every year they are supporting my program. I'm very grateful. And in the meantime, CPBR was also in the initial phase of the work. They were helping us too. Nowadays, CPBR has disappeared, and then transformation facility help us to do that transformation work, without which we cannot do anything. So here, USDA National, this is the project funded from 2013 to 18. That was very, very helpful to conduct our transgenic studies, which was very expensive and time consuming, and without any worry for doing experiment. It was very cool period from 2013 to 2018. Now I am struggling, but we are still having recruiting one postdoc. So currently for new projects on traditional mapping and identifying molecular markers. I think that staking project is going well. So maybe we will also continue on working on characterization of these genes. Anyway, with that, I'd like to thank you all for listening, and I'm more than happy to answer any question you may have.
Thank you, Professor Bhattacharya, for your wonderful talk and huge information. It will help in the future scientific community, and also your lab is still in very productive ways running. So, and we are very feeling proud to have you with us. And
thank you.
Thank you again. And now it is time for interactions. There are a lot of questions I see. Some of the students who are enjoying through the online versions, they have put some questions, and I request Vinod Bihari sir for encountering these questions and answer session.
Thank you.
Yes. Thank you, sir. So it is a very nice talk, and also my works have been included in this talk, and it's like very nice one. So people already have written their question in the Zoom platform. So I am trying to see like those questions. So one is asking, is RT-PCR data sufficient to determine the fold change of gene expression? I mean, how you determine the down-regulated genes.
Oh, how do you, okay. The question is that how do you determine the down-regulated genes?
Is it RT-PCR sufficient?
RT-PCR will give you information. I mean to say, if you have, basically RT-PCR and sequencing, transcriptomics, and for a single gene, RT-PCR is sufficient because you compare with the control. And I can understand that maybe we don't go, it's a biology, right? So we don't go minus. So you cannot have down regulation beyond 0. So when you do RT-PCR, if there is down regulation, worst will be that there is no detection. That will be 0 level in the treatment. And in the control, if you have also 0, then question doesn't arise. Then it's not down regulation. Control has to have some value, say it is 1. So if it is 1 in control and say 0.01 in treatment, that is down regulation. So we should be able to use qPCR, quantitative RT-PCR, for determining the down regulation of individual genes. If you want to do global change, then of course you have to do transcriptomics by sequencing the transcript. Does it make sense?
Yes.
Okay. If the person is asking about RT-PCR, there is also quantitative RT-PCR you can do if you don't have facility for quantitative PCR using some Tuckman assay or some fancy PCR machine like I showed here in my presentation. Many of the cases I have shown only RT-PCR product. So you can always change the number of cycles to make it more sensitive. When you do 40 cycles, then you do see amplification event from small transcript. So you really need to do multiple experiments, one for say 10 cycles, 20 cycles, 15 cycles, 25 cycles, 30, and so and so. So basically,
yeah.
But I mean to say, if we do that, then at certain stage, you will see the differences between the two.
So one question from YouTube channel I am getting. So that is there any crosstalk between the death occurred by the biotic and abiotic stress, and whether overexpressing plants showed any abiotic stress tolerance?
Yeah, that's what was the thing. Abiotic stress. We particularly have not done any experiment except for salt. The reason for doing that was also serendipity. We are checking some transgenic lines for salt tolerance for another project. And then I asked her to include that. Michelin did that. And she said that there are some tolerance to salt. But we have not published anything and we have not done rigorous experimentation. So we have done 2 or 3 times and it was consistent. But yeah, so then we did see the transcriptomes for certain genes that are involved in abiotic stress. So abiotic stress is transcriptomic experiment was useful to predict what can be the phenotype. So therefore, I think we did it some time ago, but I mean to say better than late is okay. But I mean to say based on that transcriptomes, we can design now experiment to investigate further for drought or cold tolerance, etc.
Yeah. Thank you. So one more question from Amol Solanke. Is this gene similar to glufacin gene?
Similar to what?
Is this gene similar to glufacin gene?
No, it doesn't have homology. It's a novel gene. And actually, as I mentioned earlier, we did search and there is not much similarity. This gene is pretty much, I can say, legume specific. So many legume homologs were found, and it doesn't have any homology to any domains of any other proteins. There is no domain. So basically, we have a set of few genes that are in the database that are showing identity to this protein.
So the last question for today is like, what are the homolog of GMDR1 chitin that induce marker genes of the salicylic acid and jasmonic acid mediated defense pathway, but suppresses the defense pathway regulated by ethylene? Why?
Could you repeat that question?
Yes. So what are the homolog of GMDR1? Chitin induces marker genes of salicylic and jasmonic acid mediated defense pathway, but suppress the defense pathway regulated by ethylene. Why?
Why? What are the there are many questions there. Can you just give me one?
Yeah. So what are the homolog of GMDR1?
There are 4 there are a total of 4, GMDR1, 2, and 3, and 4. And we found that only DR1 is, the expression level is low, first of all. And DR4 expression level is not detectable. And those are the 4 homologs. They have identity between them. I don't exactly know, maybe 60, 70%. But those 4 genes, we had to do qPCR, but nothing happened in 4. And what is the next question?
Chitin induces marker genes marker genes of the salicylic acid and jasmonic acid mediated defense, but suppress the defense pathway regulated by ethylene. Why?
Oh, that's in the paper, I think. The person can go to the paper. I don't know, remember the markers in name or number. It's all described in that paper. We did check for jasmonic acid as well as salicylic acid and also ethylene.
Right. So the one more common question from Zoom and YouTube are coming. Like, is this gene tested in other leguminous plants or non-leguminous plants?
Only leguminous. Non-leguminous plants include jute.
Yes.
I forgot?
Cotton.
Jute, cotton, and poplar, maybe. Yeah. No other species. No wheat, no monocot, no Arabidopsis, nothing.
They are asking, like, is it tested in other plant, like let's say rice or maize or anything?
There is no sequence because a rice genomic sequence we check for sequences, and all the genome sequence available in CBI. When we blasted it, and we don't see any similarity to any rice genes or wheat genes or corn gene, peanut genes. And peanuts, peanut has it. It's a legume. So yeah, other than legumes, there is no similar to any other crop species.
Yes.
So I don't know whether we can overexpress in Arabidopsis and so that it can activate the pathway. So that's also one of the goals we plan to do. So it may be interesting to see whether this protein can also create transgenic in rice or any other crop species, because it is possible that maybe this part is missing and the rest of the pathway is there. So we don't know. We have to explore more. Yeah.
So one small question is there, Dr.
Mhm.
Amrita Panda. How transmembrane and intrinsic proteins can be discriminated on the basis of protein sequence data?
How
The
we predict.
transmembrane and intrinsic proteins can be discriminated on the basis of protein sequence data.
There are software available. So predict Signal P is one of the, I think, Signal P is one of the program you can use. And if you put the sequence and run it within a minute or so, you'll know what is the possibility that your protein is going to go to chloroplast or plasma membrane. And but again, that is just putative. You cannot claim. So we have to do experimentally prove it. So you have to localize it into the membrane. GFP is like I showed you, GFP fusion can be a good one to monitor where it goes.
Yeah. So I think so we covered all the questions both from Zoom and YouTube for today, and I really am thankful to Professor Bhattacharya for accepting the invitation and delivering very nice lecture. And I
Thank you.
hope everybody has really learned a lot. So based on the non-hostile resistance, plant immunity, and how really that can be applied directly onto the various crops, and they also have been tested in the field condition, which is probably going to also come to the farmers for their use so in the coming future. And the similar technology can also be extended to other crops, so if anybody might be interested. So and it's very nice topic.
Yeah. I encourage to, if you have a laboratory of transformation, so I encourage to try to overexpress in rice or wheat, any crop species you are working on, and see what happens. You don't need to infect. All you need to do a small transcriptomic study for NVLRR genes to see if they are induced by this, so similar to we did. And that is less work than characterizing transgenic lines, because characterizing lines for infection, etc., it takes time. You don't know what you're getting. But I think, again, since it is omics, I'm going to support transcriptomics. And
Yes.
I think do some transgenic work, and then with GMDR1, and then try to see if any transcription differences with the non-transgenic. That could be first step. And yeah, it will be good. We'll be happy to collaborate or provide you all the sequence information, gene, etc.
Yeah, I think
So,
so. If
go
I
ahead.
think so, if there is no more question, so we will conclude this meeting for today, and we'll be again live tomorrow 9:30 sharp. And again, it will also be streamed in YouTube live. So, and thank you all for joining today. Please be in time. So, and we'll be seeing each other with a new talk. I hope so. All the circular have been reached by to everyone, and the links are also working. What little bit problem was there in the YouTube? First of all, a little, maybe 5 or 10 minutes gap was there, but lately it was live, and 60, 70 people actually watched in YouTube, and
Okay.
questions from there also were addressed. So thank you, Professor Bhattacharya. So we are really grateful.
Yeah. Thank you for organizing it and inviting me. And it's a good opportunity to see you again. And yeah, I think I just want to say thank you to all organizing committee again and for the opportunity. And it is taking the advantage of this pandemic. And I think that we can actually continue our profession without any interruption. So it's it's much easier to go to research